Welcome everyone to this week's edition of the Extracellular Vesicle Club. This is a weekly event that is endorsed by the International Society for Extracellular Vesicles. And we've been running this event since the beginning of the COVID pandemic. Today we have a very special edition of the club. We're going to Marco Brucale, who's working at the CNR in Italy. And we're going to be hearing about atomic force microscopy. And specifically, atomic force microscopy as it is applied to both extracellular vesicles and lipoprotein particles. So those of you who've been following along with the EV Club over the last month and more, you know that we've already had one club on lipoprotein particles recently, and we're probably going to have a few more articles because there's so much interest in how these particles might be interacting with EVs, masquerading as EVs, and we're going to hear today about how we might be able to distinguish those particles. So before I hand this over to our guest speaker, I would like to just encourage everybody to put your comments, your questions into the chat box here, and we will get to those comments and questions at the end of the presentation. We'll also allow you to unmute and to show your cameras then. So we look forward to a robust discussion. But first of all, Marco, thank you so much for joining today. And I know that you have some group members and colleagues who are here on the call as well who might join in the discussion. So I'd like to hand it over to you, though, and invite you to start sharing your screen.
Okay. So, thank you so much, first of all, for having me today and for the nice opportunity to share our work with the EV community. And the paper I will be presenting today isn't published yet in the traditional sense, but you can find a preprint of it on bioarxiv at this address encoded here. And the title really says it all, I think. In the paper, we explain how it's possible to use AFM microscopy to get a glimpse about the compositional content of complex mixtures in samples containing both EVs and lipoproteins by means of imaging and nanomechanics. Okay, so I don't feel I need to introduce EVs and LPs to this audience, of course, especially since I'm not a biologist, but let me spend a few words on both AFM and nanomechanics. So, as many of you probably already know, AFM is a member of the larger SPM or scanning probe microscopy family. Scanning probe microscopy just means that you have some sort of probe that you scan on a surface, then you record the interaction of this probe with the surface, and you can reconstruct a topographical map of the interactions. In AFM, the probe is simply a very, very sharp tip, and the best commercial tips available have a curvature radius of just a few nanometers at the very apex. And the interaction that we are mapping in AFM is just short-range attraction and repulsion, or with a less complex name, touch. So the most common way of operating an AFM is just doing a line-by-line raster scan of the surface, much like in an LP player, if you want. And by means of that, you can reconstruct the morphology of the surface you are scanning. This, for instance, is an example of an AFM image. It's 5 by 5 microns, this particular one. And these are EVs deposited on a glass cover slip functionalized with PLL. The second most common way of using an AFM is to use its probe as a nano dynamometer. That is, to use the probe to either push or pull an object deposited on the surface. And by doing this, you can record how much force you need to deform it, and which gives access to understanding, basically in simple terms, how soft or stiff it is. So in the specific context of EVs, AFM has disadvantages, of course. For instance, you need to immobilize your EVs on a surface. No surface, no AFM. It's as simple as that. And it's also a very slow technique in a sense, because to collect one image like this, the average commercial AFM will probably need 10 to 15 minutes, let's say. But it also has advantages, of course. The first one is that you never have to dry your sample at all. You can use your sample as you right at the end of the pipeline of the EV enrichment. So you can keep your sample in any buffer or medium you happen to have used. You don't need to add any stains or dyes or labels. You can use it as it is. And it's one of the very few ways you have to access to the direct mechanical measurement of individual particles. And perhaps most importantly, you also have access to quantitative morphology. What do I mean by quantitative morphology is that AFM images are not just fancy pictures. They are real 3D reconstruction of the actual shape of the objects on the surface. So you have actual physical lengths attached to both X, Y, and the vertical axis here, which is reconstructed as a fake color scale. So, and how do you actually measure vesicles in particular that you have deposited on a surface? Well, you might be tempted to just measure the diameter that you see here on the plane, but that's suboptimal because all the XY measurements you get in AFM are the convolution of the real sides of the object plus the sides of the AFM tip, as you can see in the cartoon. So this, of course, becomes negligible when your analyte is perhaps 100 nanometer or tenths of nanometer large, if the tip is one of the sharpest available ones, but it's still important in the case of very small dots, okay? There is a way around that people doing measuring, for instance, metal nanoparticles usually employ, which is to use the vertical measurements. As you can see here, you get no convolution on the vertical measurements as depicted here. And if you are sure that your sample is a rigid sphere, you can just say, okay, that's the diameter of my object, and I can measure it like this. Unfortunately, this doesn't work with vesicles. Why? Well, we have a hint here. If you follow the height profile of vesicles, once they are deposited, they invariably are almost perfect circular arcs, okay? That means that in 3D, what's happening is that each vesicle is interacted with the surface. It gets deformed by adhesion forces, and it ends up being an almost perfect spherical cap. But not just any random spherical cap, but rather one that has the same surface area as the original vesicle. So you just need two geometrical parameters to calculate the surface area of this object here. And then you can calculate the surface area of this sphere, which was the unperturbed vesicle, and you can trivially calculate the diameter of the vesicle. This assumption of a conservation of a surface area is reinforced by biophysics, because we know that at room temperature, at 1% stretch of the bilayer is already sufficient to rupture it. So all the intact vesicles will most likely have conserved their surface area. So with AFM images, you can automatically get the two parameters you need to get the surface area of the spherical cap and thus of the vesicle. But what's better is that if you do images in which you have dozens or hundreds of particles which are not laterally adjoining, of course, because that would impede their correct measurement, then you can reconstruct a whole distribution of diameters of the particles from a few AFM images. But of course, the first question is, how good are these? How do these size distributions that you get from AFM compare to other techniques? Well, Okay, we had the luck of being in the consortium of this very nice European project coordinated by Professor Paolo Bergese. And in particular, we had the nice opportunity of having this very same EV batches isolated by several labs and sent at the same time to the lab of Professor Deborah Berti in Florence, who did cryo-EM on the vesicles, and the very same batch was also sent to us to do our AFM morphology on that. I find it's very hard to argue with cryo-EM because you can see the bilayer of each vesicle, so I'm really convinced that all that gets into the size distribution is just intact extracellular vesicles. And also measuring the diameter of vesicles is trivial at the cryo-EM because the 2D shape you get in the image is just a direct projection of the objects. So with a simple circular fit, you can reconstruct a distribution of the diameters of the vesicles. You hunt for around 100 of these vesicles in the images and you collect them here in this histogram, okay? Then we did the same on the very same batch with AFM. And as you can see, the shapes obtained with the morphological calculations I described before gave an almost overlapping distribution. The interesting thing is that cryo-EM and AFM is very consistent in determining the diameter of vesicles, but very often, most often, they are in disagreement with other bulk techniques such as NTA. Perhaps we can talk about that later for the reason of this. Another thing that is of interest is that invariably you never find vesicles smaller than around 30 nanometers, because from biophysics you can calculate that the strain induced on a bilayer, if you try to force to close upon itself at diameters lower than 30 nanometers, it's so high that it just collapses. So no intact vesicle of below 30 nanometers of diameter can exist at room temperature. So there's a potential problem, however, as I told you, with cryo-EM, you just see, you have such an ultrastructural richness in your images that you cannot mistake an intact vesicle for something else. In AFM instead, typically, let's say your everyday experiment, you don't have the sufficient resolution to really tell this is definitely an EV. It's just a blob, a globule, and we could mistake this for something else. But we have one trick left up our sleeve. We can use nanomechanics with AFM. So we might try to assign a biological identity to this blob by using its mechanical behavior. So what do I mean by mechanical behavior is simply how the object responds to an applied mechanical stimulus like an indentation force. So to give an example in the macroscopic world, you can take an inflated balloon, push it against the wall with your finger, you can measure the distance that you are indenting it, and at the same time the force you need to indent it this far. If you plot both in an XY, on XY axis, you get a linear response, a linear increase of force versus distance. This is highly specific of two things in nature: ideal springs, which are not very abundant in nature, and intact, elastic, inflated vessels. Why am I telling you about balloons? Because believe it or not, EVs behave as nano balloons. So we have nanonewtons and nanometers now, and not newtons and meters, of course, but if you push them against the surface, you get a linear increase of force against distance, which is, as I said, very uncommon, and which can be employed to decide that, yes, this blob here was in fact an intact vesicle. What's more, you can assign a number to the slope of the linear increase in force. And the slope here will tell you how stiff the vesicle is when you measured it. In literature, this quantity is with a distinct lack of imagination, is called stiffness, okay? And you can measure it for each single particle you have in your image. So for instance, of course, we don't use fingers this time, we use the AFM probe. This is a simulation of what happens when the AFM probe, it's a coarse-grained Martini simulation, and it correctly predicts that the stored mechanical energy of the system linearly increases. So how can you use this fingerprint of intact vesicular deformation? You can use it, for instance, by pushing on an assigned globule here, and you can say, okay, I got a linear increase. This is an intact vesicle with high stiffness. This two here is also has a linear increase, so it's probably an intact vesicle too with a different stiffness. If you, of course, indent on an empty space, you don't get any signal. And if you indent on a globule which is not distinguishable from EVs on the basis of size or shape, you can tell by indenting it that if it does not have that linear increase in force, it does not have a stiffness, it probably is not an intact vesicle, which is nice, but it's also, as I told you, very, very slow. You probably need probably 20 to 30 minutes per individual EVs to do these measurements. So as you can imagine, is not something you ideally use in everyday lab practice. So is there any way out of this? Yes, we hypothesized that the shape of a deposited vesicle is influenced by their stiffness. So using the same cartoon as before, when you capture your vesicle on the surface, adhesion forces try to deform it, to make it flat, while the vesicle resists with its stiffness, okay? And so you will end up with a higher spherical cap if the vesicle is stiff, and a more flat, more oblate spherical cap if the vesicle is soft. How can you measure in a linear and robust way the shape of these vesicles? With a contact angle, for instance. So, and the contact angle can be calculated with the same parameters we already used for calculating the surface area of the vesicles. So the hypothesis is that vesicles showing a high contact angle in AFM images will be very stiff, and vesicles with a low contact angle will be softer. How can we verify this hypothesis? Well, we took a whole panel of synthetic liposomes done with our friends in Florence, the lab of Professor Deborah Berti, and we measured the stiffness of them all. Then we also measured the contact angles of them all, and we plotted one against the other, noticing that there is a distinct linear correlation between the two. So we can say that the contact angle we measure in AFM images is linked, is a representative of, broadly speaking, of a vesicle stiffness, okay? So when you move to EVs, you get for each individual vesicle these two parameters that you use to calculate both the contact angle and the diameter in solution. You can plot them in the same scatter plot by putting the sizes on the horizontal axis and the contact angle in the vertical axis. And what's best, you can, of course, do it for every putative EVs you get in your image and reconstruct a whole cluster of points, each of one of which corresponds to one of your vesicles here. If these are indeed intact vesicles of one type, most probably they will have the same stiffness. So if that's so, you should end up, as it happens with purified EV samples, with an horizontally elongated clusters of dots which show a narrow distribution of contact angles across all the spanned size range. Okay, all the cleanest samples show this behavior. Instead, if you have contaminations, you can detect it immediately by doing this scatter plot, because you will have deviations from this behavior. Here, this additional cluster, which by the way disappeared once a new batch was produced, corresponds to something which does not have the mechanical behavior of intact vesicles. Okay. So to sum up, if you see an horizontally elongated cluster of points, that's probably indicative of having one type of intact vesicles with the same stiffness across all dimensions. For a while, we were happy with this because it allowed us to detect heterogeneity in a sample. For instance, this is just a cell culture centrifuged at low speed, and you always end up with a plethora of different objects. Some of them you can rule out them being EVs just by looking at them. But even if you just plot the ones that qualitatively are compatible with being vesicles by means basically spherical caps, you end up very often with something which, like this cluster here, which cannot possibly be EVs because it crosses the 30 nanometers threshold we talked about earlier. But in general terms, we can just detect heterogeneity. We can say, okay, sorry, but in this sample, you also have something else in addition to EVs. It has this average dimension, this general size, but I don't know what this is, okay? Except that for a specific class of samples we started receiving, that is samples enriched from blood, we started noticing a striking similarity between samples produced in different labs. Okay, here we have 4 superimposed scatter plots from samples enriched from blood from 4 different labs at 4 different times. They are either from serum or plasma. They are either centrifuged at low speed, or the second one at high speed. The third one is just the most populated fraction of the size exclusion chromatography column. The fourth one has both UC and size exclusion. And as you can see, all of them are very similar. If we zoom in on this portion here, we can see that we have this region here, which is well described, which is compatible with vesicle-like behavior, but we also always have a very abundant cluster here with sizes centered around 20 nanometers, 25, which we already know from geometry that they cannot be vesicles because they are too small. And we also have this, sometimes we have and sometimes we don't, this mysterious cluster here we've never encountered before. So all the samples looked very reproducible and very similar. Now I have to do a series of caveats on this scatter plot, because since we know that these are not vesicles, all the stuff I've talked about earlier is not applicable, strictly speaking, okay? We don't know if they have a stiffness, we don't know if they conserve their surface area once they are deposited. So we cannot really say that these diameters and contact angles are not artifactual. They probably are, but since they appear, these particles reproducibly appear in the same region of the scatter plot, this means that even if the values are artifactual, there must be an underlying reproducibility and consistency of morphology and mechanics, okay? An additional caveat is that, as I told you, at these very small size, tip convolution becomes non-negligible. And again, this will perturb the determination of both contact angle and diameter. But if we ensure that we always use the same tips with the same geometry, then again, even if the values themselves we accept that they might be artifactual, they will be consistently artifactual in a way that depicts the reproducibility of the analyte morphology and mechanics. So, of course, what is the first thing that comes into mind when you have a reproducible large amount of particles in blood-derived samples? It's lipoproteins. Lipoproteins and EVs are often co-isolated because they have overlapping sides or density distributions. So enrichment methods based on only one of the two, and very often even both of them, fail in isolating totally EVs from them, okay? This is compounded by the fact that, especially in blood, LPs are way more abundant than EVs. We are talking about 10 to the 6 HDLs, individual particles for each EV as an estimate in blood, okay? And so all this concurs towards the co-purification of vast amounts of LPs. And to add the insult to injury, very often it's very difficult to even notice that you have LPs, especially if they are so small in your sample, especially if you use bulk methods, okay? So our reasoning was that, okay, but perhaps we can use nanomechanics to differentiate between LPs. So what we did was to acquire a panel of different commercially purified LP samples. And first of all, we subjected them to a series of bulk characterizations. Our colleague Marina Kretic in Milan took care of all this. And I am afraid that I cannot really go into the detail of this, but all the expected results were collected in terms of the expected markers, in terms of the protein-lipid ratio, and also in terms of the zeta potential, which was reproducibly negative for all the LPs. And one thing to notice, for instance, is that NTA wasn't able to reliably track the smallest LPs at all, okay? So once this check was made, we went on with in singular characterization of individual particles, starting with cryo-EM and AFM. We first measured the size distributions. Again, you can collect several cryo-EM images of all the LP subtypes. If they are not perfectly spherical, we did a trick of averaging to geometrical values, but okay, that's probably too technical, let's skip this. But you end up with a size distribution measured with cryo-EM. Then you do the same with AFM using the same procedures I outlined before. And what you end up is this. So you get almost overlapping distributions of sizes from both cryo-EM and AFM. This is a zoom of the most interesting part, and as you can see, they are very similar. This was somewhat surprising because, as I told you, it was not a given, it was not assured that the same procedure would work with the LPs, but it seems to just give the same diameter distribution as cryo-EM. So, however, size in itself is not able to distinguish the various LP subtypes. As a kind of game, I didn't have access to an EM image of plasma, so I stole it from this paper by Newland, which came out Newland et al. This summer. And as a sort of game, I imported it in ImageJ and just measured the lateral diameters. Even if we know that these are distorted diameters because it's deposited on a surface, then you get a distribution of sizes which already looks familiar because you get a peak here around 20, 25, and then of course larger objects. Let me also point out that I wasn't able to really automatically take into account all the small particles because they are too faint. But it's not a problem with the image. Even the best EM images, the smallest LPs don't have enough resolution, and they are so small that it's very difficult to automatically count them in. So we are probably underestimating the size distribution, the abundance of very small LPs here. Okay. So with size alone, we can detect heterogeneity. We can detect that there is a peak of probably LPs around these sizes here, but we cannot really distinguish between them. So our hypothesis was, if the different classes of LPs have different nanomechanical behaviors, then we might be able to distinguish between them, okay? So what we did was, first of all, we started with EVs as a reality check, let's say. We took EVs from purified by our colleague Lucio Barile at Cardiocentro Ticino. And these are human cardiac progenitor cell EVs, which are the smallest one. And instead, in addition, EVs obtained from red blood cells by our friends in Brescia, the lab of Professor Paolo Bergese. And as you can see, all these EVs have the expected mechanical behavior of having an average CA value that is conserved across all sizes. And at cryo-EM, all the EVs always show, as I told you before, this high contrast boundary, which corresponds to the bilayer and probably also to decorating membrane proteins, okay? So we can block out this region of the scatter plot and assign it to the most probable place where EVs will end up showing, okay? Then what we did was to start with an empty scatter plot and then add individual lipoprotein samples one by one. So the first one are chylomicrons. As you can see, they mechanically are much softer than EVs, and that's very convenient because the sizes are completely overlapped with them. So we wouldn't be able to distinguish between them if we were not measuring their stiffness. Also, interestingly, they have the same behavior as vesicles in the sense that they have a conserved contact angle across all sizes. And puzzlingly, they also have a high contrast boundary at the cryo-IM, okay? The next LP sample was very low density lipoproteins, which mechanically behave in the same way as chylomicrons. They have the same stiffness. They also have an horizontal cluster corresponding to the mechanics of intact pressurized vessels. They have an overlapping, at least in our commercial samples, they had an overlapping side distributions with chylomicrons. And they also show a puzzling high contrast boundary at the cryo-EM. The next one were LDLs. Here, for the first time, we are well below the threshold of possible putative EVs because we are always under 30 nanometers. And also for the first time, we get a mechanical behavior which is definitely not the one of intact vesicles. The cluster is also almost vertical, or should I say clusters, perhaps, because there seems to be two clusters here. And for the first time, also, we don't have a high contrast boundary at the cryo-EM, okay? The IDLs end up coinciding with the second cluster we shown in LDLs, suggesting that perhaps they were not totally purified. In this case too, the stiffness is not conserved across sizes. These are definitely not vesicles, and they also don't have the high contrast boundary in cryo-EM images. Last, HDLs, they ended up in a totally unexpected region of the scatter plot. So there are several caveats here. First of all, HDLs are so small that AFM tip convolution will definitely have an impact, okay, as I told you before. So it's not a given that since they have a lower contact angle, they have softer. It's not, we cannot conclude that, okay? And the other problem with HDLs is that they are so small that in a mixed sample, we probably lose most of them, as it happened in the EM image before. But there is another problem with that, is that they are so concentrated very often that if you need to obtain individual discrete objects to measure them, you need to dilute the sample so much that all the other species disappear, basically. So HDLs are highly problematic, but at least they always end up in the same region of the scatter plot so that we can recognize their presence, okay? So this is all the scatter plots from LPs superimposed, and we can assign each region tentatively to a biological identity in this way. And we checked what happened. The lab of Marina Kretic in Milan took platelet-free plasma from an healthy donor. They centrifuged that, and we know that this usually results in the co-isolations of EVs and LPs. And when plotted on this map, we were quite happy to see that almost all the individual particles were assigned to one of the pre-plotted regions we had, okay? You might notice, as I told, that the HDL cluster seems to be truncated, and that's because in a real mixed sample, the smallest one get lost, basically. But at least you can see that they are here. So with one scatter plot like this, what we can say is that the sample is heterogeneous. We can assign a tentative identity to the components of the sample, but perhaps we can say something more. So for instance, you might notice that this cluster of lipoproteins here seems to have more dots than EVs, for instance. So is that meaningful? Is that representative of what you actually got in the sample? Well, it is, provided that you assume that all of the objects that you had in solution at equilibrium reached the surface. How can we check if this is a reasonable expectation? Well, by taking a mixed sample and doing successive dilutions, and check that the surface density of particles at the AFM gets reduced in the same way. So there seems to be a strong correlation between surface density on AFM images and concentration in solution of the original sample. So it seems reasonable to assume that the same happens also in mixed samples. And so you can simply count the dots and say, with the usual caveat that HDLs could be a lot underestimated, you can tell how the relative abundances in terms of number density of all the species in the sample. But we also have access to the geometry of each individual particle. So we have access to their surface area. So if you are interested in the exposed surface area of your sample, you can recalculate the relative amount based on that. So surprisingly, EVs, they were 8%, if I remember correctly, of the individual particles, but they exposed 74% of the total area of particles in the sample. We can also calculate the volumes, and in this case, EVs reach 95% of the total volume of the particles in the sample. So that's basically all. To conclude, as an academic remark, to the best of our knowledge, these results represent the first single particle mechanical investigation of lipoproteins. It's very crude at the moment, much more work is needed. But from the practical point of view, following this method, you can detect co-isolation of LPs in blood-derived samples. You can assign non-EVs to specific subpopulation, and you can quantify the relative amounts of particles in terms of the number density, surface area, and cargo volume. And the method does not need any particularly advanced skill or equipment. It's quite fast, it's labor-free, so we think it's a nice addition to the pipeline of isolating EVs. The current limitations, the largest one, are linked to HDLs in the ways I explained before. And let me conclude by thanking all the team behind this. A special mention to Andrea Ridolfi and Laura Conti, who did most of the AFM measurements. Our friends at the SciTech of Milan, the group of Marina Kretic, who took care of all the wet lab and the bulk characterization. Our EV experts in Brescia, the lab of Professor Paolo Bergese. Another special mention to Jacopo, who did all the cryo-EM measurements in the group of Professor Deborah Berti in Florence, and to our friend Lucio Barile, who provided cardiac EVs. And as a last remark, let me thank you for your attention, and I will gladly answer any question you might have.
Very good. Well, thank you so much, Marco, and to you and all of your collaborators on this interesting work. The slides were amazing. I really enjoyed the slides and your presentation of them. And I think these data are really valuable for the field. We do have some questions that have come in the chat box. And I just want to remind everybody, if you haven't asked your question yet, please just put it in the chat box and I will get to you. We are allowing unmuting now, so please be ready to ask your question when I call on you. But I think I want to just start with a question of my own. You know, there have been some discussions, especially recently, about how small an EV can actually be. Do you think that your data can definitively tell us, give us an answer to that question, and also about the size distribution?
You mean the minimum size of an intact vesicle?
Of an intact vesicle, yes, indeed, yeah.
Well, ergonic considerations from biophysics tells us that if the bilayer is made by, let's say, standard phospholipids, then the strain induced by curvature below 30 nanometer is unsustainable at room temperature. It will just come apart, basically. So that's kind of hard.
And that is consistent with your data, then.
Yeah. When you measure when you are sure that all the particles in your distribution are intact vesicles, then you never have anything below 30 nanometers. Yes. Mm-hmm. Yeah, in both cryo-EM and AFM.
Yeah, yeah, and there was some interesting discussion at the recent ISEV X Tech meeting, and one of the attendees there is actually here today. So, Michael Skliar, you have a question here about the reconstructed size and whether we can distinguish here. So please go ahead and
Marco,
ask your question.
I think it's just a great talk, and I think there's just so many takeaway messages for the community. That one of them is that the vesicle size is so much smaller than the mobility size that people measure with NTA, at least 30% smaller, if not 50. I think it has very important implications, and I just have to say, I just enjoyed. It's just a great talk. Just one small suggestion is, I read your paper and I recall that I had some thoughts how robust is contact angle measurements are, and I thought that it's an easier way to look at it is just to look at the sizes when you assume that the volume is conserved versus when the area is conserved, which of course are two assumptions, right? And even though you give cryo-TEM that confirms that area is conserved, is probably, there's some deformations there. So there's probably some changes. But if this is a rigid particle, those two should be the same. So that gives you a measure of rigidity that is more robust in calculation than, maybe I don't understand exactly how you calculate the contact angle, but that is just a small suggestion that I had that really inconsequential, other than my observation, how important your talk really is.
So thanks a lot. Thanks a lot. If I understood correctly, you are suggesting to calculate sizes based on the conservation of volume?
And an area, and just compare that. And
yes, we did that.
The closer they are together, that gives you perhaps a better indication of the stiffness than calculating a contact angle that may not be calculated as robustly just because of the experimental things.
Okay, well, in fact, we checked a lot of possible ways of calculating the deformations and the sizes, and I can only tell that the one I presented today is the one that most consistently gives the same size distributions as cryo-EM, and there is a geometrical reason for using contact angle versus other sort of numerical descriptor. And that is simply because it's more consistent when for what happens at the threshold of 90 degrees. Because if you think about how AFM works, when you have a particle, and you go with the tip on the particle, if the contact angle is higher than 90 degrees, the tip won't go below the particle. So in fact, the lateral sides change meaning at 90 degrees. And the only geometrical descriptor which keeps the same meaning is the contact angle. So that, but yes, I mean, thanks a lot for the comments.
Thank you.
Great. All right, let's move on then to Oscar Boyajan.
Hello, thank you very much for this talk. Very interesting. I have my own AFM right here. I can't wait to try it myself. But just I had a question. So when maybe when we run flow cytometry on vesicles and we have those dot plots, we usually use silica beads to know where the EVs will sit on those plots. I was wondering if here you use some kind of calibration beads or calibration liposomes maybe to know where your EVs are supposed to sit in those dot plots.
Yes, we did in the first paper. It's Andrea Ridolfi et al. Analytical Chemistry 2021.
If you think, I can add the link in the chat to the paper if you enjoy.
What we did is basically to take a panel of synthetic liposomes and see that they all have the same general behavior, but at different contact angles. And we also checked what happens when you have non-vesicles like metal nanoparticles and so on, and those end up in a totally different region of the scatter plot and totally different behavior. So you cannot mistake, let's say, rigid particles with vesicles. Yeah.
Thank you very much for your answer, and
good.
Francisco for the link. Thank you.
Thanks a lot.
Thanks, and thanks, Francesco, for putting the link here. Next, we have a few questions from Pietro Parise.
Hi, Pietro.
Ciao. Impressive work. I'm really amazed. I have just two technical questions, because I think that maybe the imaging mode can influence the results of the reconstructed size and also the contact angle. So what are your feeling about it, and what do you suggest as a standard procedure to use? Have you checked different modality and turned out to be the same results, or?
What we checked was that, okay, this is very technical to AFM, I think. But if you use contact mode, it doesn't work because you destroy the sample. So you have to use intermittent contact, and the one we use most is peak force. Okay, and we tried to use different applied forces, and nothing seems to change, really. We can always use, however, the lowest possible applied force and the highest possible gain to perturb the shape of the vesicle as little as possible. We think this gives the most consistent results.
Okay. Did you try to compare intermittent mode, like with the peak force, with the oscillating one, like a tapping?
Tapping.
Because there you have the amplitude of oscillations.
No, we didn't. No, we didn't. We prefer to use peak force because you guarantee that you are not hammering the particle. You can.
Okay. Yeah, this just to as also for the audience, because if you are using the amplitude of oscillation that is typical of most AFM apart the Brucale ones, then you can end up with very strange results if you try to apply the same procedure.
Yeah,
so
okay. Nowadays,
it can be a caveat, I mean, on that. And then the last graph was amazing, with the graph for the sample with from the plasma. So, but how many images do you need, and what is this typical resolution that you need to use for getting this? Because since you're measuring also the HDL, that are very, very small. So I mean, I'm thinking that you are using also a pixel size that is quite small.
Yeah, of course. The smaller the objects you want to measure, the highest resolution you have to use. That's for sure. The morphology in, let's say, reconstruction procedure itself is very robust. We described it in the supplementary information of the first paper, and the only danger, there are two things that you need to be sure of. First of all, that you don't saturate the surface. All objects must be relaxed, individual objects with the shape they want to have without talking to nearest neighbors, okay? And the second one is that you need to have a sufficient XY resolution in the plane to not get quantized circular fits, okay? If you get quantized circular fits, you end up with having a quantized scatter plot. And that's of course not true, that's artifactual, but it still gives you a hint, generally speaking, of where they will end up when you do the images at higher resolution. You also asked about the number of images you need. It depends, basically, because it depends on how many readable objects you get in each image. So, but it's still doable within let's say in a good day, you can perhaps be measuring 2 samples, okay, a full scatter plot of 2 samples by doing, I don't know, 10 images per sample, perhaps. It's totally doable.
Okay, thank you.
You're welcome.
Thanks, Pietro. Let's go to Zukai Suo next. Question about stiffness.
Well, great talk. I want to ask you, so I think I remember you show a slide to use a liposome as a control. Do you see any difference between the stiffness between the liposome and EVs?
Yes, the liposomes were prepared to have different stiffnesses. So all of them clustered around the different values of both stiffness and contact angle. What I can tell you is that EVs in most of the samples that we happen to have measured have a surprisingly narrow distribution of stiffnesses usually. It seems like somehow they tend to always have the same kind of mechanical behavior, which was also seen with other techniques. Basically, if you read the papers by Dan Forschelen et al. In the lab of Professor Heisbaute, they do a very fine, very they painstakingly measure the bending modulus of the bilayer of natural EVs, and they also get surprisingly similar results for intact and non-pathological EVs. So perhaps it has a meaning, perhaps not, but yeah.
Also in the field, some people they discovered their EVs sometimes have multiple layers rather than just one lipid bilayer. Have you looked into a different type of EVs yet?
Yes, what I can tell you is that in when we had access to cryo-EM images, multi-layer EVs were quite rare. I don't want to say non-existent because they were there, but quite rare. There was one LP class which reproducibly and consistently had a lot of multi-layered samples, but I don't really understand that at the moment, which are chylomicrons.
Okay, thank you.
And we spotted examples of cryo-EM images of chylomicrons also from other labs having that appearance. This thing I really cannot explain at the moment. It seems strange.
Very interesting. Well, we are coming to the end of our hour here, but I do have one more question for you and anybody else who cares to chime in here. And that is about one of the very hot topics in the field right now, namely the EV corona. So some call it the protein corona, but of course it can be composed of much more than just proteins. What can AFM tell us about the associations of EVs with other small particles or with other with additional layers that may be very loosely associated?
In some cases, if you manage to go to very high resolutions, you can actually see the corona. So that's the first thing I can say. And of course, that's even easier with cryo-EM. So in a few specific cases, we actually saw the corona. I think one of the slides I shown, there was quite distinct corona in the cryo-EM image. About the association, I don't know really, because because what we do is usually, as I told before, is to hunt for isolated objects. So it's something we could try to look into in the future, but at the moment I don't have a quick and dirty answer, let's say, for this.
Good. Well, that's a fair response, and thank you for that. I think that we've all seen a lot about the capabilities of this technique today, and I certainly have a newfound respect for it and for the work that you and your colleagues are doing. So, so thanks very much. And I'd like to thank everybody else who's joined today, including those of you who are in the bar there. So thank you for coming in too.
Ciao a tutti. Grande Marco.
There you are. All right. Very good. Well,
representing us.
Yes, it's very good to have you here, and hope everybody has a good rest of the week. Hope there aren't any big disappointments for anyone with the World Cup. It was nice that they scheduled around EV Club this week. I was very disappointed.
It's not something.
Yeah, I was very disappointed last week that they had scheduled against the EV Club, and so I complained about that, and so they changed the schedule for this week. So that was very nice of them. So anyway, thanks everybody.
And you had not to pay too much attention, not to overcome with Italy's match.
Okay.
It was not too hard.
All right. Okay. Well, thanks again, and take care, everybody. Hope to see you again.
Ciao, Ken.
Ciao, ciao.
Ciao, ciao a tutti.